The next speaker that we have for this section is Slobodan Davidovic. Two different pollinator species, Apis mellifera and Bombus terrestris, as indicators of heavy metal pollution near the mining complex in Bor, Serbia.
Hello, good morning. My name is Slobodan Davidovic, thank you for the introduction, and today I'll be presenting the research that we've conducted considering the two different pollinator species, Apis mellifera and Bombus terrestris, as indicators of heavy metal pollution near the mining complex in Bor, Serbia. So, as we all know, environmental pollution with heavy metals is becoming an increasing threat because of the increasing industrialization, increase of agricultural land, urbanization, excavation, mining excavations, and all these things actually lead to the fact that the heavy metals that are deep underground can get into the soil, and from the soil they can enter the food chain, and thus they present a hazard considering the ecosystem health as well as the human health, because for that food at the end ends up on our tables. Thus, it is a growing problem, and it requires a constant monitoring and a constant innovation and rethinking how we could actually, in the best way, monitor this environmental pollution. And standard procedures that rely on monitoring stations with instruments are time-consuming and expensive, and we need to develop something like less expensive and something that will give us information in a fast pace. Now, honeybees and other pollinators with similar biology present perfect bioindicators of environmental pollution, and they have been used as such for a long time already, due to the facts that of their life style and how they come into the contact with many different environments. And the idea of using insects as bioindicators is not new. Actually, it originates from the 30s when it was just discussed theoretically, and then 30 years later, already a first study that used honeybees as bioindicators was published by Svoboda. And in this paper, the authors actually used honeybees to detect the level of contamination with the radionucleotides in the environment. So what makes honeybees and other pollinators such good bioindicators of environmental pollution? Well, honeybees, first of all, they can cover wide area in search of food up to 7 square kilometers. And in some other occasions, when there's scarcity of food near their apiaries, they can cover even greater land up to 10 square kilometers. And during that activity, they actually visit many different environments, which actually makes them suitable for checking the level of contamination or pollution in both rural and urban areas, fields, meadows, forests. Even the lakes and rivers can be, the pollution of lakes and rivers can be assessed using these insects. They can be effectively bred in large numbers, and they can be provided in unlimited supplies, which actually makes them really ideal for this type of the research. And because, like, we know that the tradition of beekeeping is spread all over the world, so it's really easy to come to the honeybees or the hives that you can either buy or rent from the beekeepers, or in other occasions, agencies or institutes that are dealing with the environmental pollution have their own apiaries. And what makes them also really good is that the hives can be positioned almost everywhere, and it also covers this wide variety of environments that the honeybees can actually test. And the honeybees can be used to test both air, soil, and water pollution, because, like, they come into all these contact with all these mediums. For example, the air, there are a number of research that showed high correlation between the air pollution and the accumulation of the pollutants on the bodies of the honeybees. The soil, well, indirectly we can assess the level of contamination in the soil, because the plants that grow on the contaminated soil accumulate the heavy metals, and through their pollen and nectar, honeybees come into the contact, they bring back to the apiaries. And water, just like any other animal, honeybees also need to collect the water, which can be used to assess the level of contamination. So now we've decided to test this hypothesis, actually, to use the two different species in the region of Bor and Majdanpek, because it represents the biggest mining area in Serbia, where copper, gold, and silver have been excavated for a really long time. So these towns are located in the eastern parts of Serbia. This is where Majdanpek is located. This is the region of town of Bor. And since the extensive mining was conducted ever since 1903, so more than 120, or actually 120 years already, you can see that around town of Majdanpek and Bor there are huge mining shafts, both underground and on surface, and a lot of tailing pods and ore tailings, which tells us that there have been enough time for these heavy metals from deep underground to come into the soil and presumably into the food chain. These are the materials and methods. So we did the sampling in June during the peak of the activities of the pollinators. As mentioned, we sampled two species, Apis mellifera and Bombus terrestris, and we've decided to sample the insects directly on the locations which we wanted to test for the pollution, or actually, yeah, contamination of the heavy metals, because we know that the honeybees that detect good pasture, or actually a lot of flowers, the same bee tends to return on multiple days. So we know that if we collect these insects on the flowers in this location, we can connect those bees with the contamination in that locality. We also had kind of a control, so up to 4 hours young honeybees that shouldn't be in the contact with the contaminated environment. So we use that kind of as a control. And on the field, we've collected 10 to 15 honeybees in order to extract RNA, because we plan to analyze the level of methylthionine expression. For the analysis of the accumulation of the metals in the dry matter of the honeybees, we've collected 25 to 50 honeybees per locality. We also collected 10 to 25 bumblebees for the same type of the analysis per locality. And as I said, we've collected them on the flowers. So most of these bumblebees and honeybees were collecting pollen or nectar on Salvia nemorosa, which also we have also collected from the same localities, and we plan to analyze the level of heavy metals in the root, trunk, and the flowers. We have the data about the soil composition, and we've tested 7 different localities. So 3 presumably uncontaminated, 1 in the faraway region of Serbia. So 2 are in the vicinity of Majdanpek and Bor, 1 in the another part of northern Serbia, and 4 presumably contaminated locations. And we tested for 8 different metals: iron, copper, zinc, cadmium, lithium, lead, gold, and nickel. And these are the localities that we've sampled. So this is the first locality near Borsko Lake. It's a location that's considered uncontaminated because it's far away from any industrial or mining facilities. The second location, Cerovo, was located near the ore tailings, so presumably contaminated. Third location is here near Todorov Potok, two ore tailings. This is maybe the most kind of creepiest location because it's next to the Krivevska River that flows out of this tailing pod. And interestingly, this meadow that we've sampled is also near the fields that are used for crops. So you're going to see how it looks like. And we had this fifth locality that's considered uncontaminated, high up in the mountains, near the vicinity of the town Majdanpek, but away from any mining or industrial facilities. Sixth location, or meadow, was located next to the copper rods factory. And seventh locality, as I mentioned, we had one remote location in the northern part of Serbia. So this is near Fruška Gora region, rural area without any mining or industrial facilities. And these are the results of the accumulation of the heavy metals in the dry matter of the bodies of the honeybees, presented here in the white, in the blue color, and the bumblebees in the red color. And we can see that the pattern of accumulation is similar in both species. And of course, as expected, the young bees that were not in contact with the environment had the lowest levels of iron, cadmium, and zinc. The uncontaminated locations, 7 and 1, had a similar level of accumulation of copper and lead. Interestingly, copper, which is an essential metal, varied in the level of accumulation for the bumblebees, while for the honeybees it was pretty much constant, which could be the result of the species-specific response. What we found really kind of unexpected is that the amount of lithium in the young bees, but also it was high level in the bees from Slankamen. These young bees are also from Vojvodina region, so it could be connected with the amount of lithium in the soil, because we know that in Slankamen, from the same locality, we've detected the high level of lithium as well. The lead was highest in the first locality that was considered to be kind of uncontaminated, which is also kind of unexpected. And for the nickel, pattern was kind of similar between the honeybees and the bumblebees, and the highest level was detected in the fifth locality. We also checked the correlation between the accumulation of metals between two species, and we've detected it for the non-essential metals like cadmium, lithium, and lead. The correlation is really high. And for the essential metals, the high correlation is of the accumulation between two species was detected for the lead. As we've seen, the copper had this kind of all around the place pattern, which can be detected here as well, because it has relatively high negative correlation of the accumulation between Apis mellifera and Bombus terrestris, while zinc and nickel had relatively kind of mild, or I can say low correlation of accumulation between two species. So in conclusions, both species accumulate metals in similar manner, which tells us that we can use them to assess the level of environmental pollution with the heavy metals, both species, not just the honeybees. And accumulation of heavy metals, especially non-essential, is high in high correlation among two species, while the accumulation of essential metals like nickel and copper exhibits different patterns, which is possibly species specific. And I would like to thank the members of the department and our head of the staff, who also all participated in the collection of the samples and the analysis of the results. Thank you all for your attention.